With the same topic. In the previous video you have seen that if the walls of the small intestines are smooth from inside rather than these finger like projections, that is called as villi. Okay, so the liquid food will stay here, it will get accumulated here and will get slowly absorbed if it has to be absorbed through the smooth walls rather than these finger like projections. So the food will slowly get absorbed in these walls only, and rest of the liquid food remain lying over here for a longer period of time without getting absorbed, and the food will only get absorbed slowly and slowly in these smooth walls. Okay, अब suppose that if I am just showing you that if we'll be trying to find out the area where this liquid food is getting absorbed if the inner walls of the small intestine are smooth. So suppose that, just for a small example, we don't have to go into that much deep. I'm just telling you that if suppose that it needs to be get absorbed in this area, so suppose that it is getting absorbed, and we have just find out okay, if we'll try to measure that particular area, length of this area, and if we measure it, it will come to be this much. So food will get absorbed at that much place only. Okay, rather than that, most of the food will remain lying inside the small intestine without getting absorbed. If suppose that these villi are present, so suppose that these irregularities of now these villi, I am trying to measure the area of these villi, the length of these villi, you will find that it will come to be this much in the same place, in the same lumen. Because of these irregular, because of these villi, the surface area, or more specifically I should say that because of the presence of these villi, the food will get absorbed here also, everywhere, like this, everywhere it will get absorbed, rather than this type where it will only get absorbed at this place. Okay, but now because of the presence of villi, the food will get absorbed all over, everywhere. That means we will be trying to find out the length of where the food get absorbed. It will come to be somewhat bigger than this one. This is small area, this will be the bigger area. Okay, so the food will get absorbed in the larger area. So what is the role of this villi? The role of the villi is to increase the area of absorption of this liquid food, so that very quickly and in larger quantity the food get absorbed in the body, and from here thousands of blood capillaries will arise, and the mouth of these blood capillaries will be inside this villi, and the liquid food slowly will get absorbed in our blood. So what is the role of these villi we have just understood today. Okay, the role of villi is to increase the surface area of absorption so that more and more digested food get absorbed in the blood. So how the small intestine is modified to increase the surface area of absorption? So it has been modified that the inner walls of the small intestine is modified, or it is having numerous finger like projections, that is villi, rather than smooth. If suppose that if it is smooth, the area is less where the food get absorbed. So I hope students that you could be able to understand what is the modification which is present in the small intestine in order to increase the surface area of absorption. Thank you.